In this module, we'll be helping you to revise physiology and transport in animals. We'll be covering the following topics: why organisms need mass transport systems, the structure and function of the mammalian heart, the pressure and volume changes in the heart during its cycle, the structure and function of blood vessels, the loading, transport and unloading of oxygen as shown by the oxygen dissociation curve, the relationship between blood, tissue fluid, lymph and plasma, the control of breathing and heartbeat, energy and exercise, including energy sources and muscle fatigue, the transport of water, minerals, and the products of photosynthesis in plants, and xerophytic adaptations of plants. Remember that as organisms get larger, their surface area to volume ratio gets smaller. This means that their cells cannot receive oxygen for respiration or remove carbon dioxide from respiration by diffusion alone. There have to be specialized surfaces for the gaseous exchange to take place, and then a transport system to carry the oxygen to cells and the carbon dioxide away from cells. The gaseous exchange systems, for example lungs and gills, are well adapted for their function and are able to maintain a steep concentration gradient. Let's concentrate on mammals now. The gaseous exchange surface is the lungs, and during ventilation, air is brought into the lungs. There are millions of alveoli to increase the surface area for gas exchange. Each alveolus is surrounded by blood capillaries. The walls of the alveoli consist of one layer of squamous endothelial cells, giving a short diffusion pathway for the gases. The endothelium of the capillaries is also just one cell thick. Red blood cells are very small biconcave discs with no nucleus, mitochondria, rough endoplasmic reticulum, smooth endoplasmic reticulum, or Golgi apparatus. This leaves more space in the cell for hemoglobin molecules. The small size and biconcave shape of the discs increases the surface area to volume ratio for diffusion of oxygen into red blood cells. In the lungs, the partial pressure of oxygen is 14 kilopascals. The air entering the lungs is around 20% oxygen, and it mixes with residual air, giving air in the lungs with around 14% oxygen. The atmospheric air pressure is 100 kilopascals, and this pressure is exerted by the mixture of gases, each gas exerting a pressure proportional to its percentage of the mixture. Hence, if the air is 14% oxygen, the partial pressure of that oxygen is 14 kilopascals. In the lungs, the hemoglobin in red blood cells in the capillaries has a high affinity for oxygen. Remember that hemoglobin is a globular protein consisting of 4 polypeptide chains, each one associated with a heme group containing an iron atom. Each of the 4 iron atoms can combine with 1 molecule or 2 atoms of oxygen, so that each hemoglobin molecule can combine with 4 molecules of oxygen. The first molecule of oxygen has to diffuse into the hemoglobin molecule to attach to its heme group, so this loading is a little slow. When this molecule of oxygen has combined with the iron atom in the first heme, it causes the shape of the hemoglobin molecule to change slightly, and this enables the second and third oxygen molecules to load or attach to the iron in the heme more quickly. The fourth oxygen molecule loads slowly, as it is harder for this one to combine. This produces the S-shaped or sigmoid curve of the oxygen dissociation curve. The table shows the varying ability of hemoglobin to carry oxygen by showing the percentage saturation of hemoglobin at different partial pressures of oxygen. The graph of these data shows the sigmoid curve. Hemoglobin can pick up oxygen at the lungs and unload it at the cells. we can see that the hemoglobin is almost fully saturated with oxygen in the lungs and stays very highly saturated as the partial pressure of oxygen drops to 8 kilopascals. This means that blood arriving at respiring tissue, such as muscle, is still very highly saturated with oxygen. Remember that red blood cells themselves do not use oxygen, as they have no mitochondria. At respiring tissue, where the partial pressure of oxygen is usually between 4 and 8 kilopascals, the oxygen readily dissociates from hemoglobin, so that a lot of oxygen is dissociated during a short drop in partial pressure of oxygen. This graph shows that when the partial pressure of carbon dioxide increases, as occurs at respiring tissue, then the curve shifts to the right. This is called the Bohr Effect, or Bohr shift, named after Christian Bohr, who discovered it in 1904. You can see that the hemoglobin unloads oxygen at higher partial pressures of oxygen, or more readily than usual. So, the Bohr shift means that the respiring tissues receive more oxygen. When the oxygen dissociation curve shifts to the right, then the hemoglobin's affinity for oxygen has been lowered. A small amount of carbon dioxide from respiring tissues dissolves in blood plasma. The majority of it diffuses into red blood cells. Some combines with terminal amine groups of some of the hemoglobin molecules. But for most of it, the enzyme carbonic anhydrase catalyzes the reaction between carbon dioxide and water to form carbonic acid. This carbonic acid dissociates into hydrogen ions and hydrogen carbonate ions. These hydrogen carbonate ions diffuse out of the red blood cells into the blood plasma and are carried to the lungs. The hydrogen ions displace oxygen from oxyhemoglobin by combining with hemoglobin to form hemoglobinic acid. This means that the oxygen is being more readily released from hemoglobin to the respiring tissue, and the hydrogen ions are mopped up by hemoglobin. This prevents the pH of the blood from becoming too low, so hemoglobin is also acting as a buffer and helping to maintain the pH of the blood at around 7.2. When the blood reaches the lungs, then hydrogen carbonate ions diffuse into the red blood cells, and the reactions just described happen in reverse. Hydrogen carbonate ions combine with hydrogen ions to form carbonic acid, which form carbon dioxide and water. The carbon dioxide leaves the blood, enters the alveoli, and is breathed out. The fetus obtains oxygen from its mother's blood. In the placenta, the blood of the mother and of the fetus are very close, and substances can diffuse across. The partial pressure of oxygen at the placenta is the same for both maternal and fetal hemoglobin. But maternal hemoglobin has to unload oxygen, and fetal hemoglobin has to load with oxygen. The fetal hemoglobin, made from 2 gamma and 2 beta polypeptide chains, has a higher affinity for oxygen than the maternal hemoglobin, made from 2 alpha and 2 beta polypeptide chains. This graph shows the oxygen dissociation curve for maternal and fetal hemoglobin. The curve for the fetal hemoglobin is to the left of the curve for maternal hemoglobin, showing that fetal hemoglobin has a higher affinity for oxygen. Hemoglobin is a molecule well suited for transporting oxygen from lungs to respiring tissue such as muscle. Myoglobin is a molecule well adapted for storing oxygen in muscles. Myoglobin consists of 1 polypeptide chain and 1 heme group. It can hold just 1 molecule of oxygen, so it is either fully saturated or completely dissociated. Once the molecule is saturated with oxygen, it holds on to that oxygen and will only give it up at very low partial pressures of oxygen. You can see from this graph that at each partial pressure of oxygen, the myoglobin has a higher percentage saturation with oxygen than hemoglobin does. Hence, the oxygen held by myoglobin is a reserve and is only used when the respiring tissue has a particularly great demand for oxygen. Diving mammals need to use this store of oxygen during a dive. The hemoglobin of diving mammals also shows a very large Bohr effect. When the blood has become oxygenated at the lungs, it is carried via the pulmonary veins to the heart. It enters the left atrium, passes to the left ventricle, and is pushed out into the aorta to travel all over the body. As the left ventricle contracts, it generates the high blood pressure in the aorta. This high pressure is transmitted through the arteries, the pulse, although it is smoothed by the recoil of the artery walls. At the tissues, the arteries divide into smaller arterioles and then into the capillary bed. The capillaries have walls made of endothelium that are 1 cell thick. At the arterial end of the capillary bed, the hydrostatic pressure is high due to contraction of the left ventricle of the heart. There is also a high water potential. Plasma, containing dissolved oxygen and nutrients, passes out of the capillary here, and the resulting tissue fluid bathes the cells. The large plasma proteins do not pass out as the capillary walls are impermeable to them. Nutrients and oxygen diffuse into cells. At the venous end of the capillary bed, there is a lower hydrostatic pressure and a reduced water potential, as water passes out of the capillary at the arterial end and the soluble plasma proteins remain behind. So here, much of the tissue fluid will pass back into the capillaries, water by osmosis, down the water potential gradient, and substances such as carbon dioxide by diffusion. About 10% of the tissue fluid will enter the lymph capillaries and eventually return to the blood where lymphatics drain into the blood system. At the venous end, the capillaries join to form venules and then veins. Veins return blood to the heart. Remember that their walls are thinner than those of arteries, as blood in veins is at lower pressure. They have the same 3 layers as in artery walls, but the middle layer, the tunica media, is thinner, having fewer elastic fibers and muscle fibers. The semilunar valves in the veins ensure that blood does not flow backwards in them, and contraction of skeletal muscles around the veins pushes the blood forward. The blood from tissues returns to the right atrium of the heart, passes to the right ventricle, and then into the pulmonary arteries to the lungs to be reoxygenated. Mammals have a double circulatory system, a pulmonary system and a systemic system. The heart beats around 70 times a minute, and the sequence of events that make up one beat is called the cardiac cycle. As the atria fill, they are relaxed, or in diastole. The valves between atria and ventricles are open a little, and some blood trickles into the ventricles. At this stage, the whole heart is in diastole. The thin walls of the atria are easily distended and offer little resistance to the blood flow. When the atria are full, their walls contract, atrial systole, generating enough pressure to force the blood into the ventricles. Semilunar valves in the vena cava and in the pulmonary veins prevents the blood passing back into them. About 0.1 seconds after the atria contract, the ventricles contract, ventricular systole. The volume of the ventricles decreases and the pressure in them increases. When the pressure in the ventricles exceeds that in the atrioventricular valves are forced shut, and as the pressure in the ventricles increases more, blood is forced out into the aorta or pulmonary vein. The semilunar valves at the base of these vessels are opened as the blood is forced out. They close as the ventricles relax and pressure falls, so that the blood does not pass back into the heart. These valves snap shut as their cusps fill with blood. See how the pressure and volume changes of the cardiac cycle correlate with the diastole and systole of atria and ventricles. Let's be sure that we can correlate the events of the cardiac cycle with the graph, showing pressure and volume changes in the left side of the heart. At point A on the graph, the pressure in the left ventricle is exceeding that in the left atrium. So, the atrioventricular valve snaps shut. At point B, the pressure in the ventricle exceeds that of the aorta, so the semilunar valve at the base of the aorta is forced open. At point C, the pressure in the ventricle falls below that of the aorta, so as blood falls into the flaps of the semilunar valve, it shuts. At point D, the pressure in the atrium is above that of the ventricle, and so the atrioventricular valve opens. There are periods of time, X and Y, where both valves on the left side of the heart and also on the right side are closed. Notice the electrocardiogram trace at the top of the graph. This trace shows the electrical activity of the heart throughout a cycle. P shows the depolarization of the atria. QRS corresponds to the contraction of the ventricle, and T shows ventricular recovery, or relaxation. Notice that there is a time scale on the x-axis. The time qt is the contraction time, and the time interval between t and q is the filling time. The time from p to p shows the duration of one beat. If we divide 60 seconds by the time for one beat, we can calculate the heart rate. Here, one cycle, or beat, takes 0.75 seconds. So the heart rate is 60 over 0.75, which is 80 beats per minute. During exercise, when the heart rate increases, the contraction time remains unchanged, but the filling time becomes shorter. How is the heartbeat controlled? Well, cardiac muscle is myogenic, which means that it naturally contracts and relaxes. It doesn't need an impulse from a nerve to make it beat. Excised hearts kept in warm, oxygenated Ringer solution will continue to beat. If all the heart cells contracted at their own natural rhythms, then the heart would not beat in a coordinated fashion. There is a specialized patch of muscle in the wall of the right atrium called the sinoatrial node. This is sometimes called the SAN, or the pacemaker. When the cells of the SAN contract, they set up a wave of electrical activity, depolarization, that spreads rapidly over the walls of both atria. This ensures that both atria contract at the same time. Between the atria and ventricles is a band of tissue that does not conduct the electrical activity to the ventricle walls. However, at a point near the top of the septum is a patch of fibers, the atrioventricular node, or AVN, that will conduct the impulse to the bundle of His in the septum, down to the apex of the ventricles, and then upwards via the Purkinje fibers through the ventricle walls. This ensures a small delay between the atria finishing contracting and the ventricles contracting, and ensures that the ventricles contract from the apex upwards, which is useful as blood is exiting the ventricles from their top parts. Cardiac output is the volume of blood pumped by the heart in a unit time, usually measured in cubic decimeters per minute. It depends on the heartbeat rate and the stroke volume, the volume of blood leaving the heart at each beat. So cardiac output equals the heart rate times the stroke volume. Changes to the cardiac output are controlled by the medulla oblongata of the brain. There is a cardioacceleratory center linked to the SAN by the sympathetic nervous system, and a cardioinhibitory center linked to the SAN by the parasympathetic nervous system. These centers receive information from chemoreceptors that respond to chemicals in the blood and baroreceptors that respond to pressure changes in the blood. When we exercise, our muscular activity increases, and due to increased respiration, more carbon dioxide is produced. This lowers the blood pH. There are swellings in the carotid arteries, called the carotid sinuses, where chemoreceptors are situated. These respond to the lowered pH of the blood due to increased carbon dioxide. There are also chemoreceptors inside the medulla and in the aortic bodies of the aorta walls. These stimulate the cardio-acceleratory center in the medulla, and as a result, nervous impulses pass along the sympathetic nerve to the SAN, and the heart rate increases. This increased blood flow will remove carbon dioxide from respiring tissue more quickly. You can also see that adrenaline, produced by the adrenal glands in conditions of excitement, stress, anger or danger, will increase both the stroke volume and the heart rate, therefore increasing the cardiac output, preparing the body for fight or flight. When the heart rate increases, the cardiac output increases, as does the force of contraction of the heart, so the systolic blood pressure increases during exercise. In the walls of the carotid sinuses and the aorta are baroreceptors. These detect an increase in blood pressure and send nerve impulses to the cardioinhibitory center of the medulla. Impulses are sent from the medulla along the parasympathetic nerve to the SAN to decrease the heart rate and reduce the cardiac output. The parasympathetic nervous system also makes arterioles dilate, giving an increased volume for the body's blood to occupy, thereby reducing blood pressure. If blood pressure drops too much, the baroreceptors send impulses to the cardio-acceleratory center. This sends impulses via the sympathetic nerve to the SAN to increase heart rate and therefore cardiac output. And also, the arterioles of the body constrict to increase blood pressure. In the medulla, there is also an area that controls the breathing rate. It consists of an inspiratory center that controls breathing in and an expiratory center that controls breathing out. When the medulla receives impulses from chemoreceptors in the carotid sinuses and aortic bodies, or when its own chemoreceptors are stimulated by a drop in blood pH, then impulses pass along the phrenic and thoracic nerves to the diaphragm muscle and the intercostal muscles. This results in an increase in the rate and depth of breathing and flushing out of the excess carbon dioxide. When the level of carbon dioxide in the blood falls, then the chemoreceptors stop stimulating the inspiratory center and the ventilation rate returns to normal. When the lungs are full of air, they expand. This stimulates the stretch receptors in the walls of the lungs to send impulses along the vagus nerve to the expiratory center of the medulla oblongata. This switches off the inspiratory center and expiration occurs. This response prevents overinflation of the lungs. The increase in breathing rate during exercise will remove the excess carbon dioxide from the blood via the alveoli to the outside during breathing out. It will also ensure that more oxygen is brought into the lungs and then into the blood for delivery to the muscles to meet their increased need for oxygen during exercise. The increased heart rate and stroke volume will ensure that more blood is delivered to the muscles. The increased temperature of the muscles and the increased carbon dioxide will produce a Bohr shift and enable oxygen to be offloaded more readily from oxyhemoglobin. During oxygen deficit, when demand for oxygen by the muscle is not yet being met by the increased breathing rate and cardiac output, oxygen from oxymyoglobin will be used by the muscle tissue for respiration. When the exercise has ceased and the person is in oxygen debt, the breathing rate and cardiac output will remain high, as extra oxygen needs to be delivered via oxyhemoglobin to help remove the lactic acid that is built up and to reoxygenate the myoglobin. Let's look at how muscles get energy for contraction during this exercise. There are several sources of energy available to muscle cells. ATP. A small amount is present in the muscle cells, but this soon gets used up. creatine phosphate in the muscles can be used to regenerate small amounts of ATP. Glycogen, stored in the muscle cells, can be hydrolyzed to glucose for respiration to make more ATP. Glucose, delivered to muscle cells via the blood from the gut or from the liver, where glycogen has been hydrolyzed. Fatty acids, supplied via the blood from fat stores in the body. There is very little ATP stored in the muscle cells, and two types of respiration can be used to produce more. When there is enough oxygen available, then aerobic respiration can be used. Glucose, or fatty acids, or excess amino acids converted to keto acids can be respired aerobically, producing carbon dioxide, water, ATP, and heat. Most of the aerobic respiration occurs in mitochondria of cells, using oxygen that has diffused from blood. Exercise powered by aerobic respiration in the muscles is aerobic exercise, and this will utilize fat stores in the body. Endurance exercise, such as cycling, swimming, marathon running, or circuit training, is aerobic. When there is a lack of oxygen, such as when muscles are in oxygen deficit, ATP can be made by anaerobic respiration, using glucose but not fatty acids. The anaerobic respiration occurs in the cytoplasm of cells, and glucose is broken down to pyruvate. The pyruvate accepts hydrogen ions to become lactate, or lactic acid. Fewer molecules of ATP are generated. Exercise powered by anaerobic respiration is anaerobic exercise. This type of respiration powers sprint events, such as the 100-meter run, a golf swing, or a shot put. As the lactate builds up, the pH is lowered, and muscle enzymes cannot work so efficiently. The muscles are fatigued. When more oxygen is available, some of this lactate, a rich source of energy, is changed to pyruvate, and then respired to carbon dioxide and water. But much of it returns to the liver to be recycled to glycogen. And now, it's time for the quiz.